Yeah, hello everyone. In the next 10 minutes, I would like to talk about reprogramming the host range of our two tailocins by playing molecular Lego. And it's here that I need. It's like
Watching anything.
I need to click.
Click space.
Okay. Yeah. And first, I will give a small introduction. Then I will explain to you how to play molecular Lego by using the VersaTile technology. And then I will end with the engineered R2 tailocins I have created. And maybe some of you are already wondering what these R2 tailocins are. Well, these R2 tailocins are similar to tail bacteriophages, but they lack a capsid and the genome. And in nature, they are produced by bacteria to kill, for example, nutrient-competing strains. And prior to the killing, it are the receptor binding proteins, which are responsible for the recognition of specific receptors on a bacterial cell wall. And if we zoom in, we see they typically have a modular buildup. So we have our first building block is the anchor domain, which is responsible for the attachment of the RBP to the base plate. And then we have a second building block is the receptor binding domain, which is responsible for the receptor recognition. And a particular advantage here is that they are really specific, that we know, but they are often too specific. And to increase their therapeutic potential, our lab used receptor binding protein engineering for that. And we used it to reprogram the host range of a tailocin scaffold. So if we select a tailocin scaffold and we take the receptor binding domain and we substitute it with another receptor binding domain, then we get an R2 tailocin with an altered host range. Okay. The limitation here is that current engineering techniques are often not suitable for high throughput engineering. And the goal of my work is to overcome this limitation by the creation of a method to enhance grafting RBPs from different origins into different tailocin scaffolds. And this is where playing molecular Lego enters the story. So here I explain VersaTile. The first step of the workflow is to create all our Lego blocks. And here we have the anchor, which is compatible with the selected tailocin scaffold, and we create multiple Lego blocks of the receptor binding domains from the different sources. And then in the second step, the VersaTile assembly, we can click all these blocks together. And the specialty here is that we can do it in one single reaction in a predefined order. And so the larger your building blocks, your box of Lego blocks is, the more creations you can make in the second step. And then in the end, we will graft this VersaTile engineered RBPs back in the selected tailocin scaffold. And we validated this platform at 3 levels. So first, we took all the, all is a lot, but we took some receptor binding domains from Escherichia phages. They were targeting 7 different O-antigen serogroups. And so different colors is a different serogroup. And we successfully grafted them in the R2 tailocin scaffold. So from this, we could simply conclude that the VersaTile-driven engineering platform was working. But then we go to the second level, and the achievement here is that we created our 2 tailocins that were retargeted for the first time towards the capsule instead of the O-antigen. And this means that now the R2 tailocin is redirected towards the capsule, so it means that the RBP enzymatically degrades the capsule surrounding the bacteria. So here we did it again against Escherichia phages, against the key antigen, but we also did it for the first time against the key antigen of Klebsiella pneumoniae. And then we thought, let's take it another level. And we created a bivalent tailocin, which was active against the key antigen of both Klebsiella pneumoniae and E. coli. And here we have the RBP architecture, and we created this architecture using 4 building blocks. 2 building blocks were from 2 different Klebsiella phages. We also had a building block from an Escherichia phage, and then the anchor was from the native tailocin. And from this, we can conclude that VersaTile is also a strong tool to create more complex RBP architectures. And all this successfully engineered our 2 tailocins were validated in 3 assays. We have the survival assay, which was used to quantify the killing effect after 40 minutes. We had the growth inhibition assay to quantify the extent inhibition after 8 hours. And then we also did the classical MIC assay to determine the lowest concentration needed to inhibit the growth for 90% for at least 24 hours. And to end this presentation, I would like, I picked just 2 examples. The upper example is an engineered R2 tailocin targeting the antigen of E. coli. The lower one is targeting the key antigen of Klebsiella pneumoniae. And we did a survival assay here. So you see different acid concentration in the x-axis, and you have the bacterial population on the y-axis. And here you see with an increasing concentration, you have a significant reduction of the bacterial population, while there is no reduction seen in the lower example. And that's why I think it's important to also do the other 2, which are a bit more sensitive, because here we also look at the inhibition, because here there is a significant effect for both examples. So here you also see a reduction in inhibition. Here it's a less significant effect, but there is also a bit higher concentrations, a significant reduction. And here you have the growth curves for 24 hours. So you see it's flat after 24 hours, so no growth. And here for the highest concentration is the same. So for the upper example, we can conclude there is a strong antibacterial effect. But then for the lower one, there is still some margin for improvement by engineering. And to end, I will just end my presentation with this keynote, and that is that powerful, VersaTile is a powerful tool to play molecular Lego with receptor binding proteins domains of phages, prophages, but also our 2 tailocins. We can retarget the host range toward different cell wall receptors and species, and we can also create more complex RBP architectures, because now we selected only 1 scaffold, but the larger your Lego block, your box of Lego blocks is, the more creations you can make. Thank you very much.